Hey Priscilla, what are you working on? Hello Mom, I'm working on a lab report for my genetics lab. Want to know about it? It is pretty interesting. Sure. Well, we used to model organism, a fruit fly which is called Drosophila melanogaster in the lab in order to study the way certain traits are passed down from generation to generation. My group had two different types of flies. One group had red eyes and the other group had brown eyes. Through the mating of the flies it can be determined which allele of the trait is dominant and recessive and what kind of chromosome it lies on. Wait, why fruit flies? Those things are pests. Why would you want them in your lab? Actually, fruit flies are the perfect model organism when working the lab. A man by the name of Thomas Hunt Morgan used them in his lab and got so much information on genetics using them. We are reading a book called The Violinist's Thumb by Sam Keen, and Keen talks about how through fruit flies Morgan was able to develop ideas and provided proof for the chromosomal theory of heredity, genetic linkage, and chromosomal crossing over. Groundbreaking stuff. Wow, I would have never thought. Yes, also fruit flies are awesome because they are super easy to work with. They have a short life cycle of only 12 days, have 4 chromosomes, and they are perfectly fine with living in a small container. The fruit flies will reproduce high numbers as well. Here, look at this picture. This is the life cycle of a fruit fly. I got it off of this website, Avirotech Pest Control. So first we have an egg. It becomes a larva, looks like the maggots we commonly see. Then a pupil, almost like a butterfly, makes a cocoon, and then the adult stage, which thing it will reproduce and start the cycle all over again. And to look at them, all that has to be done is that the flies have to be fly-napped, which is a chemical that knocks the flies right out and allows us to actually go in and manipulate them. But it is super important when you're fly-napping the flies to put the small container they are in on its side so all the flies won't go in the food. It's pretty cool. Oh, okay. So why are you using these flies again? In order to study how traits are passed down from generation to generation. But in order for you to be able to study this, you have to know some basic stuff about genetics. Genes, alleles, traits, dominance, recessivity. You know genetics stuff. I remember some of this. I took a class in college. A gene is a segment on our DNA that has information for a certain characteristic or trait, and those characteristics of different alleles, different varieties, like eye color. You can either have brown eyes or blue eyes or green eyes. Yes. Do you remember anything else? Not really. Well, okay. Alleles are the different varieties like you said. And alleles can be either dominant or recessive. Dominant traits are usually physically seen. And in order for you to physically see a recessive allele, you have to have 2 recessive alleles because the dominant allele can mask the recessive allele. A phenotype is what we see, and a genotype is the set of alleles, like the code. Genotypes can be heterozygous or homozygous. Homozygous is when you have 2 of the same alleles, and heterozygous is 2 different alleles for a trait. Okay, makes sense. Anything else I need to know before you explain to me what you actually did? Yes, we use these things called Punnett squares. They are named after an English geneticist that invented them. A Punnett square is an easy way to calculate the probability of offspring inheriting a specific trait. It looks like this, where the parental genotypes are on the sides and the probabilities of the different combinations are inside the box. So from this you can calculate the phenotypic and genotypic ratios of a generation. Geneticists denote the parents as the P generation. Their offspring are considered the F1 generation or first filial, and then when 2 offspring from the generation mate, that's the F2 generation, the second filial. Wait, so you mean the F2 generation interbreed? Like brother and sister? Yes. Just take that out of your head. It doesn't matter, they are just flies. Okay, so are you done explaining? Almost. The last thing you should know is that autosomal chromosomes are any chromosomes that aren't our sex chromosomes, the X or Y chromosomes. We need to know this because there are genes on autosomal chromosomes and on our sex chromosomes as well. So like I said before, in this experiment we have flies with 2 different distinct characteristics. We had a male that was a mutant. We will call his mutation brown eye. The males had brown eyes and the females had red eyes, the wild type trait. The hypothesis was that when the male and female flies with these characteristics mated, they would produce an F1 progeny that will look like the dominant trait, the wild-type trait. And when any two F1 offsprings mate, they will produce F2 progeny with a 3-to-1 ratio. So roughly 75% of the progeny will have red eyes and 25% of the F2 progeny will have brown eyes, assuming that the gene is on an autosomal chromosome. Okay, so what did you actually do to test all this? The first week, the parental flies that were fly-napped were placed on a note card and were observed under the microscope. First, the parental flies were separated using a paintbrush by sexing by color in order to collect data. Then we set up the small containers called vials so the parental flies can have a contained space to mate and we can observe all the F1 flies. We were given one vial and in the vial we put a cap full of instant Drosophila medium and one cap full of water and then 5 grains of yeast. Then the 4 females and the 4 males were put into the container in order to mate and produce progeny. They were left alone for exactly 7 days. After 7 days, the vials were cleared. In order to clear the vials, the flies were fly-napped when they were in the vial and the live flies were the parents and they were put in the morgue, which was just a beaker full of acetone. After 14 days from the starting date, there were flies. So we fly-napped the vial and sorted and counted by sex thing by color and the data was collected. And we picked 5 males and 5 females to mate and produce the F2 generation, and the rest of the flies were put into the morgue. 21 days from the starting date, week 3, the F1 flies that were in the vial were fly-napped and put into the morgue in order for the F2 flies to progress in their life cycle and not be confused with the F1 parent flies. It is essential to flip the vial on the side when fly-napping. Then 28 days from the starting date, week 4, the F2 flies were fly-napped, the vial and sorted and counted by sex thing by color and the data was collected. After data was collected, the flies were placed in the vial. Wow! What were your results? Well, I made tables. Here are the results for the F1 generation. Out of 109 flies, all of the F1 generation had red eyes. And here is the table for the F2 generation. Out of 49 flies, all of the F2 generation had red eyes. Okay, so what does this all mean? Well, the F1 generation all had red eyes, which supported the hypothesis that the wild-type allele is dominant to the brown eye allele. From this information it can be concluded that both parent flies were homozygous for the corresponding alleles. Wild type. Biggie brown eyes. Literally, using the Punnett square, the probability that all the progeny will express the dominant trait is shown. So as you see, all of the F1 generation should show the dominant trait since the parent flies were both homozygous for their given allele to the trait. When it comes to the F2 generation, all of the flies from the F1 generation are heterozygous, and when you mate a heterozygous fly with another heterozygous fly, there should be a 3-to-1 ratio. Roughly 75% of flies should express the dominant trait and 25% of flies should express the recessive trait, brown eyes. According to the first experimental data found, the hypothesis of the 3-to-1 ratio was not supported, which raised suspicion. There could have been errors when sorting out the flies. There was a part of the lab that experienced glitches, where F2 progeny did not survive and groups had to use new vials that were made by the lab instructor. Why wouldn't there be any F2 flies? Did something go wrong? A reason why no flies or very few F2 flies were produced could have been because the vial was not placed on its side when the flies were being fly-napped. Not placing the vial on its side made all the flies fall into the food, which made it very hard to remove the flies from the vial without killing them. Okay, so if you used a new vial of the F2 generation, why didn't it support your hypothesis that there would have been a 3-to-1 ratio? Well, the reason would be technique and precision of the person sorting out the flies. Because of the number of flies that are being sorted out by one person, there is definitely room for error. Counting 49 flies and separating by sex then eye color can bring the urgency to do things quickly, which will affect the technique being used as well as the precision of the sorter. Makes sense. Well, did other groups get the same results as your group? Well, Using the class-wide data and the other lab sections data, about 21% of flies in the F2 generation expressed the recessive trait, which does support the hypothesis that the F2 generation would have a 3-to-1 ratio. 75% of flies would express the dominant trait and 25% of flies would have expressed the recessive trait. Using the class-wide data, it can be concluded that the trait was autosomal dominant. It is autosomal because the trait was evenly distributed throughout the sexes in the F1 progeny and the F2 progeny. We can conclude it was dominant because all the F1 progeny had the wild-type trait. Wow, this lab experiment is a perfect example why flies are perfect model organisms. I bet the information you got from this lab really reinforced what you were learning in genetics. It really did. From this lab I was able to see how traits are passed down from generation within a short span of a month. It was a great experience. Every time I see a fruit fly I think about the babies I made in lab. Before I go, can I see your work cited? Sure. Here it is. Lovely honey. I am so proud of you. Good night. Good night, Mom. I love you too.